now that we understand how meiosis reduces the number of sets of chromosomes in a cell by 1/2, we can now talk about the process of gametogenesis. So gametes, now what are gametes? So gametes are essentially, I guess a good definition would be cells that fuse to make a zygote. So examples of gametes: sperm, eggs, pollen. These are all types of gametes that are produced by gametogenesis in either plants or animals. So as an example here, especially to review what is a zygote, so let's say this is a human sperm and a human egg or spermatozoa and ovum. Well, there'd be 23 chromosomes in there, one set in the sperm, one set in the egg. After fertilization, we have 46 chromosomes in a zygote. So zygote is the first cell that results from fertilization of the sperm and the egg. So we have one set of chromosomes here, one set here, and two sets down here. Now, we can see how this would be a problem if meiosis didn't reduce the number of sets of chromosomes, right? We'd have more than two sets. And we will see later in the course, this is really important that this does not happen in humans. So humans are very sensitive to abnormal chromosome numbers. So if we have more than one copy, an extra copy of any chromosome other than chromosome 21, X or Y, essentially it's lethal. So if we have a third copy of any of the autosomes except 21 or the sex chromosomes, this is a lethal condition. Missing any of the chromosomes also, any of the autosomes is also thought to be lethal. So the numbers of chromosomes are very important. And by reducing the number of sets of chromosomes, meiosis forms gametes with half the number of sets that are standard for the organism. So when those gametes fuse, it restores the normal number of sets. So we're not getting increasing numbers of sets of chromosomes or just increasing numbers of chromosomes with each generation. Meiosis helps maintain the consistency between the numbers of chromosomes from generation to generation. So with respect to gametogenesis, this is the process that makes gametes, and we are going to look at two specific forms of gametogenesis. We will look at spermatogenesis, and this reminds me, so I generalized how the sperm and the egg come together in humans. At the end of this video, I will talk specifically about how the process works in humans. It's rather interesting, and it doesn't exactly happen like I diagrammed up there, but this is a good diagram to explain how the two sets come together, how the individual sets come together to make two sets of chromosomes in the zygote. Okay, again, so we are going to talk about two forms of gametogenesis, two types of gametogenesis: spermatogenesis and oogenesis, or oogenesis. I say oogenesis. Now, spermatogenesis occurs in animals, and it is the process, the gametogenesis process, that produces spermatozoa. spermatozoa. So, which is essentially just sperm. Sperm would be just the simple term for spermatozoa. And oogenesis is the process that animals use to produce ova or eggs. Okay, so let's take a look at how these processes work. We'll take spermatogenesis first. So, spermatogenesis. And there are some nice diagrams in the notes that you can take a look at, highly detailed diagrams about how we think the cells involved in spermatogenesis, where they are situated, let's say in the testes of an animal. But we're just going to take an overview look at I'm going to provide an overview of the cells involved in spermatogenesis. So the process starts. This is the nucleus. Starts with cells called spermatogonia. Or singular would be spermatogonium. And what these do is they divide by mitosis to produce primary spermatocytes. And the primary spermatocyte is the cell that undergoes meiosis. Well, undergoes the first division of meiosis. So I'm going to diagram one tetrad in here to represent, say, all the tetrads that might be present in this primary spermatocyte. So the primary spermatocyte will undergo prophase 1, metaphase 1, anaphase 1, and telophase 1, and then cytokinesis. And as a result, and now we already went over Meiosis One, we can probably predict what the products of Meiosis One will look like with respect to their chromosome content, right? So essentially, one of these things up here is going to go to one of these cells down here, and the other one is going to go into the other one. Like this. Now these are called secondary spermatocytes. And we can ask ourselves how many chromosomes are in the primary spermatocyte relative to a secondary spermatocyte. So we can count the centromeres, right? That's how geneticists determine the number of chromosomes in a cell. So we can see there are 2 up here, 2 up here, and only 1 down here, and 1 down here. And that's good, that makes sense, because remember, Meiosis One is the reductional division stage. It reduces the number of chromosomes and sets by 1/2. So each of these secondary spermatocytes then undergoes meiosis, the second round of meiosis, Meiosis 2. So I'll just diagram one of these right here going through the process, but they both will undergo Meiosis 2. So what will happen? This will undergo prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. And when all is said and done, we have what we call spermatids. So these are the final products of meiosis. And let's look at what do the chromosomes look like here. So we can see they're no longer dyads. Up here in the secondary spermatocyte, they're dyads. Down here in the spermatid, they are monads. How many chromosomes are in the monads? Are in the spermatids? Just one. One in each spermatid. How about in the secondary spermatocytes? How many chromosomes in there? Still just one, right? So this meiosis 2, remember meiosis 2 is the equational division stage. We don't see any changes in chromosome number between in meiosis 2. So no changes in between the number of chromosomes in the secondary spermatocyte versus the spermatids. Okay, great. So I hope that makes sense. So you might notice that, okay, this doesn't look like a sperm. So there's another stage here, right? So these now have to develop and undergo some sort of differentiation to make their tails and stuff like that are found in sperm. And then the mature sperm are called spermatozoa, and they develop the tails, and those are the things that swim toward the egg cell. So let me zoom out a little bit. So we only see 2 spermatids here. Each one is giving rise to a spermatozoa, or I think it's called spermatozoon. Technically, this is the singular form. So this one is going to undergo meiosis 2 also. We're going to get 2 spermatids, and each will develop into a spermatozoon. So we should get 4 spermatozoa developing from this primary spermatocyte. And so some of the questions you will see in the problem set and maybe on the exam will do things like, it will say, okay, so if the nuclear content of a spermatozoon in an organism is, let's say, 0.15 picogram, what would be the mass of the nuclear content in a primary spermatocyte of the same organism? Well, what you would do is, if you know how spermatogenesis works overall, and you know how meiosis works, well, then you should be able to see that, okay, so we're doubling the DNA content here. The number of chromosomes isn't changing, but we're doubling the DNA content from the spermatozoon cell up to the secondary spermatocyte, and then we're doubling again as we go up to here. So the answer to a question like that, given the mass of the nuclear DNA in a spermatozoon, given the mass of 0.15 picogram, the mass of the DNA in a primary spermatocyte would be 0.60 picogram. So essentially, whatever this is times 4. And you can see, look right here, it's a little hard to see, but this little monad right here represents all the DNA in that spermatozoon. Well, up here, we've got 4 of those, but it's represented as 2 dyads. Okay, so that's spermatogenesis, an overview of spermatogenesis. Let's take a look at oogenesis. So it works a little differently. So oogenesis starts with progenitor cells called oogonia. The singular is oogonium. Let's see, oogonium. And these undergo mitosis and cell division to make the cells that undergo meiosis. And the first cell, the cell that starts meiosis, initiates, in which meiosis is initiated, is called a primary oocyte. Now, I'm going to just walk through oogenesis in animals in general, and then I'll talk about how it specifically works in humans. So there's the primary oocyte here, and it is going to undergo all of the stages. It's going to undergo Meiosis One. So prophase 1, metaphase 1, anaphase 1, and telophase 1, and then cytokinesis. But unlike in spermatogenesis, the result is going to be one secondary oocyte and one what we call a polar body. So this is going to be the first polar body. Now the polar body, we can assume it dies in animals, and we don't think that it is involved in development of the zygote or anything. We think the first polar body is lost. So the chromosomes that were delivered to the first polar body, those are lost. So the set of chromosomes that goes to the first polar body, those are lost. The lucky set of chromosomes, okay, that goes this way. Again, so take a look. Meiosis One is the reductional division stage. We should see half the number of chromosomes and half the number of sets of chromosomes. And we can see that is the case. The primary oocyte, we have 2 chromosomes up there and only 1 down here in the secondary oocyte. Okay, so now the secondary oocyte, that undergoes meiosis 2. So we have prophase 2, metaphase 2, anaphase 2, and telophase 2. And then cytokinesis. So just like before, we get this sort of unequal division of, I should say, the cytoplasm. And very little goes to, let's say, what's going to be the second polar body. One of the chromosomes goes there, or one set of chromosomes goes there. But most of the cytoplasm goes to the other product of meiosis 2. And this is going to be what we will call the ova. Well, I better, you know what? Technically, this is the ootid, which then undergoes some more development to be the ovum. Ova is plural. So we can see the numbers of chromosomes did not change. which is good because this is meiosis 2, the equational division stage of meiosis. But the dyad up here in the secondary oocyte was split, so now we have two monads. One monad is lost in the polar body, and the other one is found in the ootid. So that's in general, that's how oogenesis is thought to work in general in animals. Now let's look at how the process works in humans specifically, because I think it's really interesting. So just briefly, in humans, so the oogonia, what they do is they produce millions of primary oocytes and I'll diagram one tetrad to represent the 23 pairs or the 23 tetrads in a human primary oocyte. Now, the primary oocytes, now these are all formed in the embryos before the human with ovaries is born. So even before birth, all these primary oocytes have been formed. Not only have they been formed, but they've all begun Meiosis One, and they've stalled in prophase 1. So every newborn baby with ovaries, every newborn baby girl has in her ovaries 1 to 2 million primary oocytes that have all started undergoing Meiosis One, but they've stalled. And they will stay stalled in Meiosis One until adolescence. Now, I'm not sure exactly on the details here, but at least one of these oocytes becomes unstalled each month after puberty and goes through the remaining stages of Meiosis One. So we have metaphase 1, anaphase 1, and telophase 1 and cytokinesis. And we get the first polar body, which is lost, and the secondary oocyte. Okay, so with respect to the secondary oocyte, now the secondary oocyte will begin prophase 2 and then metaphase 2, and it will stall. It will stall in metaphase 2. So we end up with something that looks like this. So that's my meiotic spindle. And that's one of the 23 dyads that should be found in this human secondary oocyte. Now that is what is released during ovulation. And if this is fertilized, only if it's fertilized, only if it's fertilized by a sperm. So here we have a sperm entering the egg. It releases its nucleus called the pronucleus with 23 chromosomes. So only if it's fertilized will meiosis complete. And if it's fertilized, meiosis will complete, and then we will have 2 nuclei. One of the nuclei will go to what we call the second polar body, and the other nucleus will be used to fuse with the nucleus from the sperm. So that's how the process works in humans, and I think it's fascinating that meiosis does not complete until after fertilization. I also think it's fascinating when, I think about my daughters. So my wife was 30 years old when my first daughter was born, she's 14 now. So it's kind of interesting to think that, okay, my 14-year-old daughter, the primary oocyte that she was derived from was 30 years old, was essentially 30 years old. So even though my daughter's only 14, man, that cell that she was derived from is 30. Was already 30 years old. So that's amazing that, this cell kind of just was stalled in meiosis one in my wife's ovary, one of her ovaries, for 30 years. Well, I think there are some theories on age of mother when a child is born, and increased likelihood for genetic disease, and maybe it could be because as these primary oocytes are stalled in meiosis one, they might be accumulating mutations. Again, those are theories. I don't know if they've how much data supports that, but it's interesting for me to think about that. These primary oocytes are stored for so long, so many years, before they're used to produce the next generation in humans. Okay, so that's it for gametogenesis, and that's it for lecture 2. So I'll be moving on to lecture 3 videos soon.